Assalamualaikum and hi. So the topic that we're gonna talk about rice plants produce an important staple food. It is crucial that this plant can thrive against stress such as drought and salinity. Explain the several strategy to improve such stress tolerance in rice plants by giving example. So the objective, we need to identify the strategies to improve stress tolerance in rice plants.
Introduction. Scientific name for rice is Oriva sativa. Rice is life. Millions of people, economies, diets, and cultures have all been influenced by rice. Rice is life for over half of humanity. So this is strategies to improve such stress tolerance in rice plants. First is breeding, then transgenic rice and crop management practices. So firstly, breeding. What is breeding? Plant breeding can be broadly defined as alteration caused in plants as a result of their use by humans ranging from unitensional changes resulting from the advent or agriculture to the application of molecular tools for precision breeding. So this is three method that I found. First is blend breeding based on the selection of observed variants, disgrading their origin, and then generation and selection of expanded variation by control mating. And the last one is monitoring the inheritance of within genome variation and selection of specific recombination. So the first one is plant breeding based on the selection of observed variants discarding their origin. So the point is primitive plant breeding. Primitive plant breeding, the earliest form of plant breeding, involved the selection of naturally occurring variants in the wild and cultivated fields. And then genetic variation underwent continuous selection pressure through food gathering or planting harvesting cycle. So the second is generation plant domestication and the domestication syndrome. So the accumulation of mutation during domestication lead to the development of the domestication syndrome, a set of characteristics that make cultivated species dependent on human for survival, and, and molecular variability in domesticated plant is often smaller than in related wild species due to the founder effect during the domestication. Um, the second one is generation and selection of expanded variation by controlled mating. So first is idiotype breeding, and the second one is population breeding. So ideo ideotype breeding aims to improve the pedigree methods capacity to promote gains for quantitative traits, especially yields, by changing simple traits that are positively correlated with them. And this method is based on the hypothesis that changing certain trait can significantly improve complex trait. And then for the population breeding involve improving the phenotypic performance of an intermating population by increasing the frequency of favorable alleles, controlling trait of interest. And recurrent selection breeding is an open-ended scheme of consecutive rounds of selection and recombination. And the third one is monitoring the inheritance of within genome variation and selection of specific recombinants, that is genomic selection and marker assisted selection MES. Okay. For genomic selection, recent advancement in genotyping technology have reduced the cost, allowing scoring thousands of markers in population under selection. Genomic selection purpose using information from all markers in a plant to predict its breeding value without evaluating its phenotype based on statistical model. And genetic selection accelerates genetic progress to quantitative traits, especially challenging traits like Yield potential. Large companies are heavily investing in high throughput DNA analysis and bioinformatics for seamless integration of genomic selection in breeding program. And then the second one is marker assisted selection (MES). MES involve in monitoring multiple genes simultaneously, allowing the breeder to build specific allele combination that maximize agronomic value. And resistant gene pyrimidine, a typical application of MES, involve combining multiple resistant genes in a variety for durable resistance against pathogen. And then molecular markers can be combined in optimized set for simultaneous genotype multiplexing, enabling high throughput application in breeding programs. Next, conventional breeding to improve such stress tolerance in rice plant. Um, first point is breeding population and selection process. The first step involves creating or obtaining a breeding population with high variability for salt tolerance. The second step include selecting individuals from the progeny that exhibit both desirable traits from the parents and a high degree of salt tolerance. The second point is two factors in determining salinity tolerance. Two critical factors are considered in determining salinity tolerance. Threshold highest permissible salt level without yield reduction, and slope percentage reduction in yield per unit rise in salt level above the threshold. The third point is challenges in conventional breeding. Conventional breeding faces challenges due to the difficult in establishing physiological character, de- defining salt tolerance, controlled by multiple genes known as quantitative trait QTL. And the second one is limited parental resources for breeding contribute to the overall challenges in achieving success. Last but not least, variation in salinity tolerance during growth stage. Rice varieties exhibit varied levels of salinity tolerance at different growth stages, with tolerance during germination, active tolerance and maturity, but sensitivity during early seedling and reproductive stages. Example: the sub-1E gene was successfully introduced into rice plants to enhance their submergence tolerance. This gene enables the plants to survive in flooded conditions for up to 2 weeks, significantly improving their productivity in flood-prone areas. The second example is Pokali is recognized as a potential salt-tolerant donor with a major QTL cell identified on chromosome 1. Lastly, FL47E, derived from a cross between Pokali and IR29, is promoted as an improved donor with high seedling stage salinity tolerance and desirable agronomic strength.
What is transgenic rice? Rice plants have been modified to increase micronutrients such as vitamin A, accelerate photosynthesis, tolerate herbicide, resist pests, increase grain size, generate nutrients, flavor, or produce human protein. How transgenic method work? The Chinese scientists put recombinant HSA protein promoters into 25 rice plants using agrobacterium. Out of the 25 plants, 9 contain the HSA protein. The genetically modified brown rice makes the same amino acid sequence as HSA. They call this protein Oryza sativa recombinant HSA. Besides, by transferring genes from the soil bacterium Bacillus thuringiensis into the rice genome, Genetically modified BT rice is resistant against the Lepidopteran pest, damage, and thus less require the amount of insecticides used. Other than that, they also use method TT511, synonym to B363, is an insect-resistant transgenic rice transformation, even harboring a hybrid CRY1AB-AC gene regulated by the rice 18-1 gene promoter and the Nopolin synthase NOS terminal. Although transgenic rice TT511 show great potential rice production, it has not yet been approved for either human consumption or safe for the environment up to now. The benefits of transgenic rice. First generation transgenic varieties have been benefited many farmers in the form of reduced production costs, higher yields of both. In many cases, they have also benefit to environment because of reducing pesticide usage or by providing the means to grow crops with less tillage. Second, increase the nutritional content, resistant to pests and disease, and tolerate to environmental stress.
Next strategies that I'll be sharing will be crop management practices. Planning and executing strategies used to maximize crop growth, yield, and quality is known as crop management. First step in crop management will be mulching. Mulching around crops helps to control temperature, retain soil moisture, and lessen the effects of the heat on plant roots. Why mulching? First, it helps control soil temperature fluctuations, reducing the stress on trees. Protect trees' roots from extreme heat and cold. Secondly, can maintain the good level of moisture in the soil where it can be absorbed by roots. This is especially beneficial during periods of drought. Last but not least, it helps to hinder weed growth and seed germination of undesired plants. And next step for crop management practices will be irrigation. Irrigation practices helps conserve water resources and promotes responsible water management in agriculture. Why irrigation? Irrigation is properly controlled helps maintain strong root system. Strong roots increase a plant's capacity to tolerate stress because they are better able to absorb nutrient and water. The availability of nutrients in the soil is impacted by irrigation. Rice plants that receive optimal nutrient uptake through proper irrigation management are healthier overall and more resilient to stress. So, we have come to an end of our presentation. Rice is a crucial food source for more than half of the world's population and is grown in 114 countries globally. Increasing rice production can have a significant positive impact on communities. In addition to its importance as a food crop, rice is also recommended for desalination purposes. It can grow well in flooded conditions, and the standing water in rice paddies can help remove salts from the topsoil, making the land suitable for other crops. Improving salinity stress tolerance in rice faces various challenges. However, there are also opportunities to address this issue, such as breeding, transgenic rice, and crop management processes.
I know